Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the another topic, that is your detection of the VIBRIO uh, parahemolyticus. Okay, so this is what this come under the category of gram negative bacteria. This VIBRIO parahemolyticus come under the category of gram negative bacteria. That means that when we do a gram staining, then it will give a pink color colony. Okay, it is a curved rod shaped bacteria. Curved rod shaped bacteria, and they are found in sea and in a when indigestion occur, then it will create a gastrointestinal illness. So this type of bacteria mainly found in a sea, and it cause gastrointestinal illness, which means that if a human being consume the food which is contaminated by this bacteria, then there is a symptoms occur, and these symptoms are what? These symptoms are watery diarrhea, abdominal pain, vomiting, nausea, fever, and blood, sometimes bloody stool. And bloody stool. So that's why it is very important to detect this bacteria into the water, because it create a symptom into a human body, and sometimes if the diarrhea is severe, then it will leads to a death of the human being. That's why it is very important to detect this type of bacteria. Now let's start the today's lecture. VIBRIO parahemolyticus, they are what? They are a gram-negative bacteria, oxidase-negative, ferment glucose without gas production, and it does not produce a H2S in TSI. TSI what? It is a triple sugar iron agar media, and it is motile bacteria which cause a enteritis in human. See, this is a gram negative bacteria that I already told you. It gave a pink color colony. They are what? They are a oxidase negative, and it ferment the glucose. And when it ferment the glucose, then there is a no production of the gas, and also it does not produce the H2S when it is treated with the TSI, that is your triple sugar iron agar medium. It is come under the category of the motile bacterium which cause a enteritis in a human being. Now come to the next slide, which is the principle of this experiment. So glucose, salt, tapol broth are used here. Okay, so this glucose, salt, tapol both are what? They are enriched in Vibrio parahemolyticus. Then this glucose is utilized by the microorganism, and the tipol inhibit the migration of the halophilic organism. See, in glucose, salt, tipol broth, there are two things. One is your glucose and one is your tipol. Glucose is utilized by the microorganism. Utilized by the microorganism, and this tipol, what they do, they inhibit the growth of the other type of the halophilic microorganism, halophilic microorganism and also inhibit the growth of the gram positive microorganism. Okay, and the incubation period time is overnight at 37 degrees Celsius, that is 35 plus minus 2, that is your 37 degrees Celsius. Inoculation on TCBS agar plate give a round green or a bluish colony. Okay, so this TCBS agar plate give a confirmation for this species, and the confirmatory is what they are round green or bluish color colonies. So this is the principle at which this experiment are based. Now come to the next category, sorry, come to the next slide, that is your culture media. We all know that culture media is what? It is a substance which is required by the microorganism for their growth. Okay, so here two type of the culture media is required. One is your glucose salt tipol broth, and the another one is your TCBS, that is your thiosulfate citrate bile salt sucrose. So first we are going to see the composition of this glucose salt tiphenol. This composition is for a 1 liter. That is means that 1 liter distilled water. So it contains peptone, 10 gram, meat extract 3 gram, glucose 5 gram, sodium chloride 30 gram, methyl violet 0.002 gram, and tipol 4 gram. Okay, so these are the composition of the glucose salt tiphenol broth, which require peptone, meat extract, glucose, NaCl, methyl violet, and tipol. And this requirement is for a 1 liter of the distilled water. If you require in 500, 200, 250 ml. So according to your need, you can adjust the amount of these chemicals. Okay, then the final pH will be a 7.5 plus minus 0.2. Then it will be autoclave at 121 degrees Celsius, temperature 15 psi, pressure for a 20 minute. Then the another type of the media that we required is a thiosulfate citrate bile salt sucrose, that is TCBS. So it required agar. Again, this composition is for a 1 liter. Agar we required 14 gram, bromo bromo bromothymol blue, bromothymol blue required 0.04 gram. Then we required a ferric citrate 1 gram, mixed peptone 10 gram, ox bile 5 gram, sodium chloride 10 gram, sodium folate 3 gram, sodium citrate 10 gram, sodium thiosulfate 10 gram, sucrose 20 gram, thymol blue 0.04 gram, and yeast extract 5 gram. Now the final pH will be 8.5. Then after that we autoclave it at again 121 degrees Celsius temperature, 15 psi pressure for a 20 minutes. So this is the chemical composition of the culture media that we required for performing this experiment. Now come to the next slide, that is your procedure. Same what we have to do, we first of all we disinfect the surface of the bottle, pouch, cup which already contain the sample with the help of the 70% of the ethanol, and thoroughly mix the sample via vigorous shaking so that it will achieve a uniform distribution. And again this will be performed into a laminar airflow. So for this you have to clean the laminar with the help of the 75% of the ethanol, and then you have to switch on the UV light for 30 minutes. Then after that switch on the airflow and then work on it. Okay, and the this surface of the bottle, pouch, cup which contain a sample will be disinfected with the help of the 75% of the ethanol, so that if there is any contamination there then it will be removed. Otherwise this type of the contamination will come into the sample and it will cause a error. And we mix it thoroughly so that the uniformity will occur when we take the sample. Now the next is your when we have a sample, then we filter that sample, and the filtration amount of the sample is according to our required volume need. And this filtration will be done with the help of the sterile membrane filter, whose pore size will be a 0.45 micrometer. And then we place this filter into a 50 ml of glucose salt phenol broth. Okay, the filtrate that we get will be mixed with the 50 ml of the glucose salt phenol broth, and then we incubate it at overnight. After overnight, we take a loop of the streak from that solution. Suppose that this is your 50 ml of that solution. We take a loop, and this loop is streaked into a plate in which already TCBS agar are there. Okay, then we incubate it for 18 hours at 37 degrees Celsius. Observation is what? Suspicious colony of V paracamelo paramolyticus on TCBS medium are 2 to 3 mm round with green or blue center. Okay, so if we get the round colonies of green or blue, then it is what? It is the required microorganism or required bacteria. Now, identification, suspicious growth of the V. parahemolyticus is confirmed through a different biochemical conformation, and these conformation are what? They are the biochemical tests are what? If there are a typical suspected colonies are there, then we select at least 5 colonies from a positive agar medium, and then we streak it on a nutrient agar plate and again incubate it at 36 degrees Celsius for 18 to 24 hours. Then after that we do a further biochemical or serological colony. Suppose that this is your TCBS agar and here we get a colonies. Now from that colonies we select at least 5 colonies, and these 5 colonies are streaked into a nutrient agar media. Then after that it will incubate at 37 degrees Celsius for at least 24 to 18 to 24 hours. Then after that, with the help of that colonies that we get on the nutrient agar plate, we do a biochemical or a serological confirmation test. Now the first test that we are going to perform is a gram staining. So again, we do a gram staining, and when we do a gram staining, we get a pink colonies, which indicate that it is what? It is a gram negative bacteria. Now the next is your test for a oxidase. We take a streak colony on a nutrient agar slant in which we are added 2 to 3% of the NaCl. Then we allow the growth for 24 hours. Then to a nutrient agar slant of the culture, we add a few drop of the test reagent. Here the test reagent is what? 1% of the alpha naphthol, which are already present into a 95% of ethanol, or the equal amount of para amino dimethylene alanine hydrochloride in water. If the reaction is positive, then it will indicate by the appearance of the blue color, in which we add within the 2 minute of addition of the reagent. Okay, so within a 2 minute of the addition of this 1% of the alpha naphthol in 95% of ethanol, of para amino dimethylene alanine hydrochloride in water, we get a blue color appearance, and it will indicate that the presence of the microorganisms. The next is your huge Leffon's test. Step a fresh culture from a nutrient agar slant into a 2 tubes of huge Lefson medium, in which we add a 2 to 3% of the sodium chloride, one of which is then overlaid with a small amount of the sterile liquid paraffin, then incubated at 37 degrees Celsius and examined up to 4 days. Acid formation show up a yellow colony in a tube without a paraffin, which indicate that the oxidative utilization of the glucose, and acid in both tube indicate a fermentative reaction. So this is about the confirmatory test. Now expression of the result, presence or absence of the VIBRIO parahemolyticus given after the confirmation of the preservative colonies in a sample examined. Test for the VIBRIO parahemolyticus is equal to present or absent per x ml of the sample. Suppose that if it is a 30 colony in 10 ml, then it is represent at a 30 CFU per 10 ml. So this is all about the today's lecture. I hope you will understand. But if you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.